Hello everyone, I'm here to present our work called Integrative Data Analysis to uncover genes and pathways underlying the overall prognosis of lung cancer brain metastasis. We know that metastasis is estimated to be the cause of about 90% of cancer-related deaths. Brain is one of the most sensitive parts that has been the target of metastatic incidences with mean survival time of about 6 months. Lung cancer is reported to be the major source of brain metastasis, in a point that some sources have reported up to 50% of brain metastatic incidents are seeded from lung primary tumors. And how rapid lung tumor metastasizes to the brain plays a major role in survival chance and treatment decisions. Due to the importance of the whole problem, there are lots of studies going on, but still there are several concepts that are underappreciated in the literature. The first thing is that the heterogeneity in the tumor sometimes is neglected. Multiple regions of analysis should be taken into account, not just focusing on lung or not just simply focusing on brain, but we have to have a holistic view. And then we should have a whole transcriptomic view, not only focusing on the genes that we are hypothesizing that be relevant to our study, because we might miss out some of the understudied genes. So the objective of this study is just to discover the molecular processes linked with the time to metastasis, while taking the aforementioned points into account, like taking the tumor heterogeneity into account and consider both brain and lung together, and not limiting our focus to a specific group of genes, but have a whole transcriptome view. So in this regard, we are using a valuable data set generated and collected in Dr. Zhang's laboratory by our friend Robert Abdo. This data has been collected using nanostring digital spatial profiler technology, and it profiles the expression level of 35 patients in the brain and lung of them. And as we know that nanostring digital spatial profiler is expression profiler really similar to traditional ones, with the main difference is that not only the expression information is preserved, but also we know which part of the tissue the expression is extracted from. For example, if we extract gene expression, if we profile the gene expression of a tissue, we know that, for example, this gene expression is extracted from a tumor cell reached or immune cell reached part of the tissue. So we have profile our gene expression of these patients in 5 different regions. 2 of them are in lung, which are tumor and tumor infiltrating lymphocytes region, and 3 of them are in the brain, which are tumor infiltrating lymphocyte regions. For short, we'll calling them TIL hereafter, and tumor microenvironments. Our data consists of 5 different regions in 2 organs of 35 patients. Not all of the expression data is available for all of the patients, so there are some missing regions for some patients. Our study is structured into 3 steps. First, we want to see what are the sources of variation in the expression data. In other words, we want to see what are the different expression signatures in the patients that causes the differences between the patients. And then we want to see what are those gene signatures or expression signatures are affecting time to metastasis. So basically, some of the some of the some of the sorry uh, some of the gene signatures are really contributing to differences between patients, but they're not because of they're not relevant to time to metastasis. Some of them are, for example, because of gender differences or age differences and so on and so forth, which are not necessarily relevant to time to metastasis. And then after discovering the gene signatures and sources of variation, which we are going to call them factors hereafter. After discovering the relevant factors, we want to see what are these factors link, what are the biological interpretation of these genes, of these expression signatures. So first, we group patients into two classes. First one are fast metastases, the ones with metastatic incidence earlier than a year, and then a slow metastasis, the ones with metastasis later than 20 months, about more than 2 years. So, and after that, we apply the method that I've just mentioned. So we first use a tool that is called multiomics factor analysis, or MOFA for short, to integrate the data between different regions and discover data factors. Factors are those sources of variations or expression signatures, whichever you are more comfortable with. And then each factor is a group of genes contributing to this same expression signature. The factors with significantly different activity level in patients of the fast versus slow groups are named relevant factors. So this is like the second step that can be seen in figure 1. Now, the factors with significantly different activity level in patients of the fast versus slow groups are named relevant factors. And after we discovered the relevant factors, we want to see the the biological interpretation of these gene signatures. So we try to perform a gene set enrichment analysis against cake database on the gene sets that are corresponding to those relevant factors, and find the pathways that are linked with rapidity of the metastasis. So you can see in figure 3, we applied MOFA on our data set, and we have discovered 15 different gene signatures, which are trying to explain a level of variation in each part of our tissue. Like, this is the lung tumor, this is the brain tumor, this is the brain microenvironment, tumor infiltrated regions, and so on and so forth. So we have discovered some gene signatures, and then we applied the second step to, and then we went with the second step to find the ones that are more relevant to time to metastasis, and then we proceeded with the gene set enrichment analysis, which the results was that the discovered pathways could be grouped in 3 categories: immunity-related, sameness, and metabolism. Immunity-related pathways like neutrophil extracellular traps and antigen processing and presentation. You can see, for example, in figure 2, that antigen processing and presentation is decreasing faster metastasis groups, but so in the tumor cells with more elusiveness. You can see here the red depicts the downregulation of these genes that are playing a role in the pathway to show to present the antigen to our T cells. So with the decreased level of antigen presentation, the tumor cells are going to be more elusive and having the more chance to metastasize. And there are some related pathways like focal adhesion and ECM receptor interactions that are that have different that suggest different levels of sameness and higher level of sameness in fast metastatic groups, and different metabolism patterns like Warburg effect and oxidative phosphorylations is observed as well. Thank you for your consideration. I want to appreciate Dr. Shusheri's and Dr. Zhang's contribution, Dr. Shusheri's and Dr. Zhang's lab contribution to our work. And I'm ready to take any questions.